After detailed detective work, experts now believe this is the only surviving piece of clothing worn by Elizabeth I.
This was actually reserved only for the immediate members of the royal family. And of course, if you're looking at sort of a late 16th century member of the royal family, that's just Elizabeth.
The announcement arrived like a rolling tremor across the scientific community, echoing far beyond the closed doors where the research team had quietly labored for years. For centuries, historians had relied on letters, portraits, and state documents to decipher the inner life of Queen Elizabeth I, the monarch whose reign shaped an era and whose persona became an emblem of power and inscrutability. Her body, however, had remained untouched by modern science, sealed in history with as much mystery as majesty. When the world learned that a team of geneticists had succeeded in extracting analyzable material believed to be derived from her preserved garments, cosmetics, and personal items, excitement collided with disbelief. Historians, biologists, and the public alike braced for revelations that might illuminate long-debated aspects of her health, identity, and ancestry. What no one anticipated was how profoundly the findings would challenge the image the world had carried of her for generations. Before we start, hit the like and subscribe buttons for more exclusive news. The project began with an unconventional approach. Direct sampling from royal remains had long been considered off-limits for ethical, cultural, and political reasons. However, advances in ancient DNA recovery techniques allowed scientists to shift their focus to the objects she handled daily. Her gloves, lace collars, embroidered gowns, and cosmetic tools, preserved since the 16th century and maintained in stable archival conditions, contained microscopic traces of sweat, oils, and skin cells. Some items even bore residue from lip products and herbal powders. The research team developed new protocols specifically for extracting genetic fragments from substances centuries old, integrating methods from paleogenomics and proteomics. The work required patience, precision, and an acceptance that many attempts would fail before any workable sample emerged. But when the first sequences began to assemble into coherent strands, the team understood they had crossed a threshold no historian had imagined possible. One of the earliest surprises arose from the mitochondrial DNA, which traces maternal lineage. Elizabeth's lineage through her mother, Anne Boleyn, had been documented extensively through Tudor genealogies. Yet the DNA revealed hints of ancestry far more complex than expected. The sequences suggested maternal markers consistent not only with the English aristocracy, but also with genetic signatures more commonly associated with populations from the Mediterranean basin. While historians had long speculated about the exotic heritage rumored in court gossip, the genetic findings suggested a deeper and more ancient blending in her maternal line. These characteristics did not change her identity as a Tudor, but they painted a picture of a lineage shaped by centuries of cross-regional movement. The implications stretched beyond genealogy. It reframed discussions about cultural exchange in medieval Europe and the interconnectedness of noble bloodlines. Even more compelling was the discovery of several genetic variants linked to physiological traits that historians had only guessed at through portraiture and written accounts. For generations, scholars had debated the monarch's reported sensitivity to perfumes, her apparent struggles with recurrent illnesses, and her occasional withdrawal from court life. The DNA hinted at a constellation of biological conditions that could explain these behaviors. One variant suggested a predisposition to severe migraines, consistent with descriptions from contemporaries noting sudden episodes in which she retreated from public engagement. Another indicated heightened susceptibility to autoimmune symptoms, which might align with the chronic skin issues she attempted to conceal beneath heavy cosmetic layers. These possibilities breathed new realism into long-studied anecdotes, turning what had once been speculative interpretations of behavior into grounded biological context. Equally startling was the data recovered from proteins embedded in the cosmetic residues. Elizabeth was renowned for her distinctive appearance, especially in her later years, when her public image became a carefully crafted emblem of monarchical power. Her white-painted face, ruby-tinted lips, and elaborate wigs were tools of political symbolism. But analysis of the residual compounds revealed chemical interactions between her makeup and her skin that suggested the cosmetics might have caused significant physiological stress. Lead-based face paint, widely used at the time, was already known to be hazardous, but its interaction with genetic vulnerabilities indicated a deeper strain on her health than previously imagined. The proteins indicated inflammation markers at levels that, in a modern medical context, would be considered concerning. These findings did not diminish her legacy, but rather intensified admiration for her resilience in the face of physical hardship. The genomic findings also reawakened debates around Elizabeth's famously enigmatic approach to marriage and motherhood. For centuries, historians had speculated about whether her refusal to marry was rooted purely in political strategy, personal trauma, or, as some suggested, a private medical condition. The DNA provided clues that added another dimension to the debate. Certain variants revealed in the reconstructed genome indicated a high probability of congenital complications that might have rendered pregnancy dangerous or even impossible. While definitive conclusions remained beyond reach, the genetic implications suggested that reproductive challenges may have been a silent burden shadowing her reign. These findings did not reduce her political choices to biology, but enriched the understanding of what pressures she may have balanced in her carefully curated public life. A particularly haunting aspect of the analysis involved the stress markers embedded in the keratin proteins drawn from fragments thought to originate from her wigs and accessories. The proteins contained signatures associated with prolonged physiological stress, malnutrition cycles, and disrupted sleep patterns. Court records frequently describe the immense weight of leadership on her shoulders and the political turmoil she navigated with skill, but the scientific evidence suggested that the emotional and political strain manifested at a biochemical level. The data implied periods of intense endocrine disruption, the kind seen in individuals carrying extraordinary burdens without relief. These biological echoes of stress painted a portrait of a monarch whose inner turbulence mirrored the tumultuous era she governed. The more the scientists studied the genome, the more they found themselves confronting the dual nature of Elizabeth's existence as both a historical figure and a human being, with vulnerabilities etched into her DNA. The recovered sequences revealed a surprising degree of genetic diversity. Although the Tudor bloodline was shaped by generations of intermarriage common among European royals, Elizabeth's genome showed fewer indicators of severe inbreeding than some of her contemporaries. This finding intrigued population geneticists, who speculated that the diversity in her maternal line may have counterbalanced the narrower paternal heritage. These results opened discussions about the hidden genetic complexities of European royal families and how genetic variation may have influenced the personalities, health, and dynastic decisions of historical leaders. Another layer of insight came from the unexpected discovery of certain immune system markers. Some suggested strong resistance to particular forms of viral infection common during the 16th century, while others indicated vulnerability to conditions that could have easily spiraled into severe illness. This delicate balance reflected the precarious health environment of the Tudor era, when outbreaks swept through court and countryside alike. Elizabeth's ability to endure long years during periods of disease outbreaks had often been regarded as a combination of luck and careful isolation. The genetic data nuanced this understanding by showing it may have been a matter not only of circumstance, but of innate biological resilience intertwined with hidden susceptibilities. The implications extended beyond historical curiosity. By examining her DNA, researchers found unexpected connections to modern studies of chronic illness, stress physiology, and environmental exposure. The interactions between heavy metals in cosmetics and genetic predispositions, for example, provided insights relevant to contemporary toxicology. The potential autoimmune markers shed light on how genetic vulnerability interacts with environmental conditions, offering a rare case study spanning centuries. In this way, the study became more than a window into the life of a monarch. It evolved into a bridge linking past and present scientific challenges. One of the most unexpected discoveries came from the study of gene expression markers preserved in microfragments of tissue protein. While the DNA gave structural insights, the protein spoke to the lived experience of the queen. Through molecular patterns indicating inflammation cycles, stress responses, and hormonal fluctuations, the researchers began to assemble a picture of a woman whose physical experiences were far more complex than her portrait suggested. Her life at court required continual performance, constant vigilance, and psychological fortitude. The molecular evidence suggested that this performance exacted a physiological toll that went far beyond the visible. More controversial still was the identification of neurological markers often associated with exceptional verbal memory, heightened pattern recognition, and rapid cognitive processing. These characteristics were consistent with historical accounts describing her sharp wit, fluency, and decisiveness. Scholars had long celebrated her intellectual gifts, but the possibility that her cognitive strengths had a genetic component added a striking new layer. It did not diminish her achievements or the rigorous education she received. Rather, it illustrated how her natural predispositions may have intertwined with opportunity and ambition to shape her extraordinary role in shaping English history. Discussions erupted among academics about nature, nurture, and leadership, all spurred by the unexpected illumination of her biological blueprint. As scientists continued to sift through the data, they encountered variants associated with traits that scholars had not widely entertained in discussions of her personality. One such variant related to heightened sensory perception, suggesting she may have experienced certain sounds or scents with greater intensity. If so, it could explain anecdotes of her aversion to strong perfumes or loud disruptions in court. While such biological interpretations remain speculative, the possibility of heightened sensory sensitivity enriched the emotional texture of her historical portrayal. It suggested a monarch attuned to her environment in ways that could amplify both her strategic strengths and her vulnerabilities. The revelations did not stop with physiology. Some genetic markers hinted at distinctive metabolic patterns, possibly influencing her eating habits and energy cycles. Historical records mention her selective diet and unusual eating patterns, and the DNA suggested a rationale grounded in biology rather than eccentricity. Variants linked to irregular glucose regulation pointed to possible fluctuations in energy levels that she may have intuitively managed through specific dietary choices. These insights added subtle detail to the understanding of her daily life, shedding light on choices that had long been dismissed as personal quirks or courtly fashion. Perhaps the most unsettling discovery emerged when researchers examined residues from her cosmetics and clothing associated with her final years. Alongside genetic material, they detected compounds indicating the presence of heavy metals and plant toxins interacting with her biological profile in alarming ways. These combinations produced biochemical markers consistent with chronic poisoning, not necessarily from malicious intent, but likely from the very cosmetics and treatments she used to maintain her symbolic image. These findings transformed the understanding of her later years, suggesting that her devotion to projecting strength and beauty in a politically turbulent environment may have unwittingly subjected her to dangerous physiological decline. The implications were profound. Elizabeth had cultivated an image of ageless authority, a living symbol of national stability. Yet her DNA and protein markers told a different story, one of a body under siege, battling the consequences of cosmetic practices and the expectations of monarchy. Her commitment to maintaining her iconic visage may have come at the expense of her health, a tragic irony woven into the very fabric of her reign. Scholars began exploring how this revelation reframed the cultural history of beauty standards and their physical tolls on historical figures. The analysis also raised questions about how the pressures of leadership in a patriarchal society shaped her body's biochemistry. Cortisol markers suggested prolonged stretches of extreme stress, consistent with the relentless scrutiny and political tension she faced as a female monarch navigating expectations that no predecessor had confronted in the same way. The molecular evidence hinted at cycles of stress so intense they may have caused physiological changes that shaped her emotional world. This did not reduce her accomplishments to stress responses, but it illuminated the demanding reality behind the steady composure she projected to the world. At the same time, contrasts appeared in certain genetic markers associated with emotional stability and resilience. These markers, while not deterministic, suggested that she possessed innate predispositions toward emotional regulation that may have fortified her against the psychological storms that accompanied her reign. Historical descriptions of her adaptability, humor, and capacity for forgiveness gained new resonance when viewed through the lens of her genetic profile. The interplay between these markers and the stress indicators painted a portrait of a leader whose emotional strength was both biologically rooted and consciously cultivated. The project deepened in complexity when researchers examined microbiome traces embedded in the materials. Though fragmentary, These microbial signatures suggested patterns of exposure linked to the environment she inhabited. They hinted at the physical spaces where she spent her time, the food she consumed, and the daily routines that structured her life. This layer of analysis opened new doors for historians attempting to reconstruct the lived reality of the Tudor court. It provided clues about her proximity to certain environments, her interactions with personal attendants, and the rhythms of her daily existence. Though subtle, these microbial insights enriched the understanding of her bodily presence in the world. The revelations far exceeded the expectations of the team that began the project. Their goal had been simply to explore whether human DNA could be extracted from historical artifacts with enough clarity to yield meaningful results. Instead, they found themselves unveiling a narrative of a queen whose body carried the imprint of her era, her burdens, and her remarkable resilience. The analysis challenged the idealized image of Elizabeth projected through portraits and political legend. It presented her instead as a woman whose physical reality was shaped by genetic inheritance, environmental exposure, and the intense pressures of leadership. These findings invited renewed discussion about the intersection of biology and history. Elizabeth had been a figure of immense political significance, but the genomic evidence reminded scholars that she was also a human being navigating the limitations and possibilities of her own body. The science did not diminish her legacy. If anything, it enhanced the sense of awe surrounding her life and reign. Understanding the biological challenges she may have faced only heightened appreciation for her accomplishments and the strength she displayed in guiding her nation through upheaval. As new interpretations took shape, the historical community became increasingly interested in how these biological insights might influence broader studies of early modern monarchy. The possibility that genetic factors shape the decisions, personalities, and vulnerabilities of rulers offered an intriguing dimension to political and cultural analysis. It suggested that the human bodies behind royal portraits may have played a role in shaping the course of history in ways previously overlooked. Elizabeth's genome became a focal point for this emerging field, a rare bridge between scientific discovery and historical interpretation. The implications reached beyond academia. Cultural conversations erupted about beauty, health, and leadership, inspired by the realization that a figure celebrated for her regal composure may have suffered physically from the very symbols of power she employed. Debates flourished about the ways societies impose visual expectations on leaders, especially women, and how those expectations carry hidden costs. Elizabeth's story, illuminated by the new genetic data, became a lens through which people examined contemporary pressures and the human consequences of projecting an idealized public persona. The findings also stimulated reflection on the nature of historical memory. For centuries, Elizabeth had been depicted as a near-mythic figure, a queen defined by her intellect, ambition, and political success. The genomic analysis introduced an unexpected intimacy, revealing details of her biological struggles and internal experiences. It challenged readers and scholars to reconcile the monumental figure of Elizabeth with the individual revealed through her DNA, a woman shaped not only by her will and education, but also by the vulnerabilities encoded in her cells. Despite the extraordinary illumination offered by the genetic data, many questions remained unanswered. The findings opened new avenues of inquiry rather than closing existing debates. They invited scholars to consider how much of history rests not only on documents and decisions, but on the unseen workings of the human body. Elizabeth's DNA became a testament to the intricate interplay between biology and destiny, a reminder that the stories of great leaders are also the stories of living organisms adapting to their environments. What the scientists uncovered inside her body was indeed shocking, not because it revealed weakness, but because it brought her humanity into sharper focus. Her genome held traces of pain, resilience, mystery, and complexity that resonated across centuries. It embodied the paradox of a queen who projected invulnerability while simultaneously enduring profound physical and emotional strain. The revelations allowed the world to glimpse not only the monarch, but the woman beneath the crown. The event marked a turning point in how historical figures might be understood. It demonstrated that the past still carries hidden voices waiting to be heard through the tools of modern science. Elizabeth's DNA stood as a bridge between eras, a biological manuscript that offered new insights into one of the most fascinating figures in history. The implications rippled out to historians, scientists, and the public, inviting reflection on how deeply intertwined biology and history truly are. Through the genetic lens, Elizabeth's life appeared both more extraordinary and more relatable. Her experiences, once filtered through the stylized art and formal language of her age, became grounded in biological reality. The findings revealed that beneath the grandeur of her reign lay a story of a body navigating the challenges of illness, stress, and environmental exposure. Her triumphs were thus not only political but personal, shaped by a profound endurance that resonated in her genetic code. If you liked this video, please give it a like and subscribe to our channel. Also, could you leave your comments below and tell us, how do you expect these new DNA findings to change her historical image? We want to hear from you. Thank you for watching, and see you next time.